Welcome everyone to another episode of the Wine Science Commentary. It's Monday and that means another paper is waiting for you. My name is Lorenz and today we'll look at fermentation temperatures and how they affect the varietal expression of your wines. So if you like the content, of course hit the subscribe button down below in the corner, and that's it from my side. I would say, let's do it. Influence of fermentation temperature on volatile thiols concentrations in Sauvignon blanc wines, published in 2006 in the International Journal of Food Microbiology. Head researcher here was Isabelle Masneuf-Pomarède, and also Denis Dubourdieu was involved in this research. So let's have a look at these guys for a second. Here it is. Isabelle Masneuf-Pomarède, Professor of Viticulture and Enology at Bordeaux Science Agro University. She is, as it says here, Professor of Enology and Head of the Scientific Department of Bordeaux Science Agro. Isabelle Masneuf-Pomarède conducts research on wine microbiology, especially of wine yeasts, including fermentative and spoilage yeasts. And what about this guy here, Denis Dubourdieu? Some of you might know him. He unfortunately passed away in 2016. He's one of the most renowned wine scientists out there up to date. And why is that? Because first of all, he consulted for one of the best wine estates in the Bordeaux area, including Chateau d'Yquem, Cheval Blanc and Margaux. He published more than 200 research papers. So him and his team, they were involved in discovering thiols, of being responsible for the main aroma composition of Sauvignon Blanc wines. He introduced skin contact for Sauvignon Blanc wines. He introduced lees steering and all that kind of stuff. So he really improved winemaking in the Bordeaux area. And some of his students even called him just Dieu, which is part of his name and means God, of course, in French. So that already proves what kind of guy he was. Okay, so let's jump to the introduction here. Low temperature enhanced the wine contents of some volatile compounds produced by the yeast during the alcoholic fermentation, such as esters, acetates, medium chain fatty acids. For this reason, low temperature fermentation between 10 and 15 degrees is of interest to winemakers to enhance the production of some volatile compounds and improve the wine aromatic profile. However, this low temperature can easily cause sluggish or stuck fermentation. Three major aromatic volatiles, 4-MMP, 3-MH, 3-MHA, were identified as responsible for the box tree grapefruit and passion fruit nuances of wines made from this variety. And of course, these volatile thiols are also found in other varieties, including Gewürztraminer Riesling, Colombard, Petit Masseng, Cabernet Sauvignon, Merlot, just to mention a few. Of course, you also find them in so-called non-aromatic wines like Melon de Bourgogne or Pinot Grigio, for instance. And the volatile thiols are released during the alcoholic fermentation due to the transformation of their corresponding S-cysteine conjugate by the yeast. So let's have a look at these thiols for a second. As you can see, there are more than 3 volatile thiols that you find in wines or the grape itself, but 4-MMP, 3-MH and 3-MHA are of course the most important ones. So 4-MMP, as you see, is the responsible odor for black currant. Very low threshold, 0.8 nanograms per liter. That means if you put a drop of this molecule in a swimming pool, you can certainly smell it. And the concentration range in wine is very low, until 400 nanograms per liter. 3-MHA is the box tree aroma. In terms of perception threshold, 4.2, so 5-fold compared to 4-MMP, but also the concentration range is much higher. Which is found in wine. And then 3-MH is this typically grapefruit, passion fruit odor, and you see here threshold 15-fold compared to 3-MHA, but also the concentration is much higher. Then what about these thiols in the grape juice and in the corresponding wine? So in the grape juice, these thiols or thiol precursors, they are conjugated with cysteine precursors, for instance, or also conjugated with glutathione in this case here. And there's also a third pathway. But however, within the grape juice, these natural thiol precursors are odorless, so you cannot smell them because they're conjugated here and they are linked to these other molecules. But once fermentation starts, yeast is able to split or to cleave this linkage here, thus liberating the thiols, and thus you can smell them. And yeah, so in the grape juice, these thiol precursors are non-oxidizable, but in the wine, of course, as we all know, they are highly oxidizable and you have to work to preserve them. And the final concentration of thiols in your wine will be approximately 10% of the concentration found in your juice. So that means if you have like 1 million cysteine 4-MMP precursors in the juice, then you end up like 100,000 in your wine. And of course, this depends on your winemaking technique, this depends on fermentation temperature, as we will see, on the yeast you will use, and so on and so forth. Now, experimental design. So in this case, they conducted 2 different experiments, which were more or less similar in their intention. So in the experiment 1, they created a model fermentation medium, which consisted of sugars, acids, nitrogen, mineral salts, vitamins, and thiol precursors. So very similar to a must of a thiolic variety. And then this model fermentation was fermented via 3 different yeast strains. They used Simaflor VL3C from Lafor, they used Win13 from Anchor, and they used the NW3 yeast, which I didn't find any further information on it. And then these different ferments were conducted in triplicates, so at a microscale of 750 milliliters at 3 different temperatures, 13 degrees, 20 degrees, and 24 degrees. And then they analyzed the volatile compounds and also some wine basic chemistry. Then experimental 2 was done with 2 different Sauvignon Blanc musts. Both came from the Bordeaux area, termed R for right and L for left. And these 2 different musts were fermented via 4 different yeast strains, again BL3C, Anchor, Win13, and Simaflor X5 from Lafor. And then they used also a hybrid yeast crossing between Cerevisiae and Bayanus, termed H9 here. So all these yeasts are very powerful in liberating thiols from the respective precursors. And furthermore, every ferment for each individual yeast was conducted in duplicate at a 350 milliliter scale, fermented at 2 different temperatures, 13 degrees and 24 degrees. Well, let's jump back to our paper. Let's look at the results here. So here you see the results of experiment 1. So you remember this was the model fermentation solution here. We have 3 different yeast strains, as I showed you before, which were fermented at 3 different temperatures. And here, this chart A represents the final concentration of 4-MMP, so of the box tree passion fruit cassis aroma. As you see here, for yeast strain A, which was VL3C, the higher your fermentation temperature is, the higher the respective 4-MMP concentration was in the vine. So you have the higher the temperature, the more release of this thiol, of this 4-MMP. The same trend is followed by strain C, whereas for strain B, which was this N3W, we have the highest concentration at 20 degrees. And then chart B shows us the concentration of 3MH, so of passion fruit, grapefruit aromatics, and you see the higher the fermentation, the higher the respective 3MH composition or concentration in the wine. So we have this clear trend here. What about acetic acid? So for strain A and B, you see the lower the temperature was, the higher the acetic acid production in the final wine. Whereas for strain C, we see that the highest concentration was found in ferments at high temperature. But for sure, we are below acetic acid threshold, which usually lies between 0.7, 0.8, something like that, depends on the taster. And so, yeah, these acetic acid levels are not very powerful in the wines. Then let's have a look at experiment 2. Let's make this a bit bigger. And as I mentioned, we have 2 Sauvignon Blanc musts, R and L. Then we have our 4 different yeasts. We have 2 different temperature levels, and then we have our parameters here. What about residual sugar? So at 20 degrees, the yeast was clearly able to ferment the wines to almost 1 gram per liter, so the wines were dry. whereas at the lower temperature at 13 degrees, the yeast struggled quite a bit, and in one case even produced a semi-dry wine with 15.9 grams per liter. So here you see the yeast struggled quite a bit at these low temperatures. In terms of tartaric acid, we find higher levels in the 20-degree ferments compared to the 13-degree ferments. We will look at this, at the reasons further below. What about ethanol levels? What about VA or acetic acid? Here we don't have any clear differences. Well, let's look at the liberation of 4-MMP, 3-MH, 3-MHA of the same experiment. Let's make this a bit bigger. And here we see, in terms of the 4-MMP concentration at 13 degrees, for some ferments the 4-MMP concentration was so low that it was not detectable, whereas at the higher temperature we clearly see it was detected in each ferment. What about 3-MH? What about this grapefruit passion fruit aroma? Here we see clearly that at 20 degrees we have the extensive amount of higher production of these 3-MH molecules than compared to the 13-degree ones. So in this case here we have a 10-fold increase, and a 10-fold increase, 4-fold increase, and so on. So clearly higher levels at higher temperature. 3-MHA follows the same trend, but not so extensively. And so you see 5-fold increase, 2-fold increase, 6-fold increase, and so on. So quite clear trend. Then one more thing I want to show you is this here. Principal component analysis was applied to the results obtained for the 7 parameters measured in wines R and L. And we see clearly that the temperature of 20 degrees correlates more with the total acidity, with 4-MMP, with 3-MHA, and with 3-MH, whereas the lower temperature correlates more to the residual sugar, so we end up with a not so dry wine and also to volatile acidity. Whereas the ethanol level is doesn't correlate with anything. It's quite in the middle here. So very interesting. Let's go to the discussion. What I say, the PCA analysis showed that 4 parameters among the 7 studied, so the 3 volatiles and total acidity, are correlated with high fermentation temperature. In our conditions, more tartaric acid may have precipitated when alcoholic fermentation is conducted at 13 degrees, thus explaining the higher level of total acidity found in wines obtained at 20 degrees. The final levels of 4-MMP and 3-MH in wines were higher when the alcoholic fermentation was conducted at 20 degrees than at 13 degrees. It was previously described that low fermentation temperature enhanced the aromatic characteristics of wines, possibly because of greater synthesis and a greater retention of volatile flavors. Our results concerning levels of volatile thiols in wines show opposite conclusions. Conclusion. The results of our study clearly demonstrate that the high fermentation temperature between 18 and 20 degrees is better for the expression of the varietal aroma of Sauvignon Blanc wines, and probably also for other varieties containing this S-cysteine conjugate, such as Graumanseng or Gewürztraminer. Fermentation temperature is inexpensive, or this fermentation temperature is inexpensive for winemakers and preferred to prevent sluggish or stuck fermentations. However, some yeast strains seem to be well adapted to follow or to low fermentation temperature. So, very interesting study. What is our take-home message? Again, you see that here at 20 degrees we had a higher synthesis of these 3 major volatile aromatics, and also we end up with a higher total acidity, probably because at lower temperature we have a higher precipitation of tartaric acid. Then, does this automatically mean that our wines get better the higher the fermentation temperature is? Well, it depends on what you want to produce. Like if you think of Sauvignon Blanc wines from the Loire, from Sancerre, from Pouilly-Fumé, these wines are the benchmarks for Sauvignon Blanc, and certainly they are very restrained in their aromatic composition when you compare them to New Zealand Sauvignon Blancs or Chilean Sauvignon Blancs. So it's a tool for you. It's something you should have in your repertoire if you want to make wines which are more, which express more aromaticness than if you want to make wines that have lower intensity of these volatile thiols, then you can use lower temperature. So it's certainly up to you to decide what to do. That was a weekly episode of the Wine Science Commentary, guys. I hope you enjoyed it. I hope you liked it as always. If so, why not subscribing to the channel? Hit the subscribe button down below in the corner. And next week I have another cold soak paper lined up for you. Highly interesting content as always. And yeah, see you next Monday. Stay safe, stay hydrated. Have a nice week.